So I am very pleased to introduce Dr. Jennifer Collinger as our neuroprosthesis seminar speaker, and she'll be speaking on the topic as you see here. Jennifer received her undergraduate degree from the University of Pittsburgh in bioengineering, and as well as her PhD. There she is now an assistant professor of PM&R at the University of Pittsburgh. And it really is okay to be an engineer and a faculty member at a clinical department. You will thrive. So I'm going to just share a couple of things about Jennifer. I was pretty impressed with, as I was looking through her CV. In 2012, she was named as one of the 10 world-changing innovators by Popular Mechanics for her ECOG brain-computer interface research. Same year, brain-machine interface research was named one of the top 10 science breakthroughs of the year by Science Magazine, and that was for the Lancet article. And again, 2013, she received the top 10 Clinical Research Achievement Award in the U.S., awarded by the Clinical Research Forum. That's pretty impressive. So we are very honored to have Jennifer join us and to give us the seminar. Jennifer, come on over.
Well, thank you to Dr. Che and for the FES Center for having me here. As I've been telling a number of people, it feels surprising that this is actually my first visit to this center. We've had a lot of graduates from here come and work with us in Pittsburgh, so I sort of feel like I have a good handle on things, but it's been great to see some of the labs yesterday, and I'm excited to meet with everyone today. So I am going to be talking about primarily our intracortical brain-computer interface work that we've been doing in Pittsburgh. And our goal is really, right now we're working on controlling a robotic arm and hand for people. So I will tell you today a little bit about some BCI user design priorities from a survey that we did a number of years ago. Then I'll tell you about the progress that we made in achieving high-dimensional motor control, some challenges that we've run into, specifically related to object interaction and manipulation. I'll tell you about some recent work with somatosensory feedback using intracortical microstimulation. And finally, I'll tell you a little bit about a project that we did in collaboration with Carnegie Mellon University, where we're combining BCI and some autonomous robotics to improve function. So as everyone in this room knows, people who have severe disabilities may be limited in their ability to communicate or to interact with their environment. And many traditional assistive technologies, such as a joystick or a keyboard, require some amount of dexterity to operate. And so that's where brain-computer interfaces can come in for some people, where we can establish a direct link between the brain and some type of external device. And we think that it has the potential to provide a high degree of freedom and intuitive control for these users. So this data was actually probably collected in 2011, but we asked people the same question that Kim Anderson had asked a number of years before us, essentially, just to characterize our population. So this was 60 veterans with spinal cord injury who were participating at the National Veterans Wheelchair Games. And we asked them which of these different 8 functions that you can see across the bottom would have the most impact on their quality of life, positive impact. And so what you can see, I don't know if, for people with tetraplegia here in blue, arm and hand function is a top priority, as well as bladder and bowel, and even restoration of walking. I think this is fairly consistent with what others have shown. Our group did have a higher preference towards walking than maybe they'd seen in other samples, but otherwise it was fairly consistent. And then for paraplegia, bladder, bowel, and walking were the top functional priorities. We've chosen to focus on restoration of arm and hand function. Right now, as I mentioned, we're still using a robotic arm for doing that. We did ask people, well before we had even started actually doing any clinical BCI trials, what would they like to control with a BCI? And so we kept it fairly simple. We only gave them a 3/4 page description of what a BCI might be and what these technologies were. But what you see, as I'm sure everyone here is well aware, the preference for people was really FES for restoring either hand grasp for people with tetraplegia or lower body function, bladder, bowel, right? And I think this goes along with their goal is to really restore movement to their own body, to make them like they were prior to their injury, to have this technology be invisible. And so, I think that is a a very lofty goal that we should try to work towards, and really happy to see what's been going on here and making progress towards that. There was some interest in controlling a computer and wheelchair. At the time, there was moderate, a third of people were interested in controlling a robotic arm. I definitely don't think that is the way forward for long-term function. A more recent survey out of Stanford has shown there was a little bit more of an increase in interest in robots. I think people are maybe more familiar with them now than they were, 6 years ago. As far as design characteristics, being able to operate the device independently was really the most important. The goal of the BCI is to reduce the dependence on caregivers, and so if they needed to have a technician or have a caregiver intervene, that sort of defeated the purpose of having the BCI. Training time was considered least important. People seemed to be willing to spend more time to learn the device if it would work well for them once it was set up. 70% of people said that non-invasiveness was very important. We asked the question in a slightly different way and said, would you be willing to consider having surgery to implant electrodes? When we asked it that way, more than half of people said definitely or very likely. I know that Jane Huggins out of Michigan has done a follow-up survey in people with ALS, and it seemed to make a really big difference whether the surgery was an outpatient surgery that you could go home from that day, or whether there was an inpatient recovery. So there may be some education component or the degree of surgery that is also important here. So now to get to the meat of the presentation, I do see so as you are probably familiar with, this is the device that we're using with a shortened wire bundle for this photo. But essentially, there's this external connector that we use to record signals from the brain. And you can see on this lateral X-ray, in our participants, we've implanted 2 of these devices. They're connected by a gold wire bundle down to this intracortical microelectrode. And you can sort of see that on the X-ray here, that there's 2 electrodes implanted in motor cortex. If you zoom in on that array, there's 100 electrodes. They penetrate about 1.5 millimeters into the cortex, and the footprint is 4 by 4 millimeters. And so we record neural firing rates, and we convert that into control signals for the robotic arm. And for most of the work that we've done to date, we're controlling that arm in endpoint velocity space. And I'll tell you a little bit more about how we do that. So our first participant was implanted about 4 years ago, and she was a 52-year-old woman who had tetraplegia. She was diagnosed with spinocerebellar degeneration. I think we're not exactly sure what caused her spinal cord injury there, because she didn't actually appear to have any degeneration in the cerebellum. But essentially what it presented as clinically was a motor complete SCI at the C4 level, and her sensation was generally intact below that level. She did have some regions of hypersensitivity, but for the most part it was intact. And she had 2 of those Utah arrays implanted into her left motor cortex. We determined where to place the electrodes based on presurgical planning. There's a lot of literature out there suggesting that there could be cortical reorganization after spinal cord injury. I think we've mostly found that the somatotopic organization that is typically seen in motor cortex is preserved, but there is at least individual variability. I don't know that we have enough subjects to say whether we've seen any complete plasticity. But essentially, if you're looking, this is the top-down view of the brain. Here's the front, here's the back. And if we zoom in on the left hemisphere, here's the central sulcus. And so we're looking at motor cortex for this participant. And we had her imagine grasping her hand, shrugging her shoulder, pursing her lips, and doing complex finger movements. And so we tried to place the arrays where you see these purple boxes, putting one array more towards the finger area and one array more towards the shoulder area, and just making sure to stay more medial of the Broca's area. A lot of our BCI work is based on things that were started to be discovered in the 1980s. And just for anybody who is not familiar with this, although I'm sure that's probably unlikely with Dawn sitting in the audience, who has done a lot of this work to actually use this information to control a BCI. So just to really briefly summarize what these experiments were, they had a monkey while they were recording data from motor cortex, 1 neuron at a time, and having them reach towards these targets arranged in a circular pattern in a 2D plane. And so this is looking at activity from 1 neuron. Everyone in the room is probably familiar with a raster plot, right? Rows are different trials. Each vertical line is the firing, is 1 action potential from a unit. And so what you're seeing is when they're reaching to targets in this hemisphere, there's a lot more activity than when they're in this hemisphere. And you can represent that mathematically as a cosine tuned function. So as you change the direction of movement, there's one direction that it fires maximally for, and in the opposite direction it fires at its minimum firing rate. And so that can actually be described by a fairly simple linear equation where the firing rate of that unit is linearly related to the velocity in those two different directions. And initially, control was done just based on summing up the activity from all of these units. So when you're moving in this direction, each of these lines is representing what that neuron is firing. The length of that line is proportional to how much it's firing. And so neurons with preferred directions that are pointing up are firing more for this target, and vice versa for the down target. And we've actually used that idea to really scale up to controlling a prosthetic arm. So we were using a robotic arm. We control it in endpoint extrinsic coordinates. So we control the XYZ translational velocity. We also control yaw, pitch, and roll of the wrist. So rather than doing individual joint control, we operate in an extrinsic space. And then we initially started out by linking all of the fingers together for a single grasp dimension. So this is that simple equation that I showed you before, where we assume that the firing rate of the units can be predicted or explained by velocity in those two directions. And along with Andy Schwartz, we thought, let's just add a bunch more terms and assume that cosine tuning kind of spans, will follow. And so this is the model that we use, where the firing rate is assumed to be a linear combination of the translational velocities, the rotational velocities, and then a grasp dimension velocity as well. And you can put this in matrix form. So for all of your units, you have the firing rate at each instant in time, and that's equal to the velocity for however many dimensions of velocity you're controlling by these beta coefficients. And we use a method called indirect optimal linear estimation to find the inverse of that beta matrix. But essentially, when we're doing control, our output is the velocity, and however many dimensions we're controlling that day is equal to the firing rate determined by these decoder weights, which are essentially an inverse of that beta matrix. The way that we train our decoder, I think, is a pretty common method, but it's based on this idea of using observed or attempted movement to get that initial mapping from firing rates into command signals. So this is really founded on the discovery of the mirror neuron system, right, where they were recording not necessarily in motor cortex, but in other areas of the brain like premotor cortex and parietal cortex from the monkey when he was doing reaching movements, and then an experimenter reached in to grab that same object, and the activity was very similar. And so that's been, very well characterized, that there are certain areas of the brain that fire the same, whether you're doing a movement or whether you're observing a movement. So a lot of the BCI work that had been done in animals was moving in that direction of trying to train the cursors without overt movement, and that has seemed to translate over very well to people with paralysis. So initially we start out our calibration by having, I'm going to show you a virtual reality version of a training for a 7-dimensional task. But we have the arm move automatically by the computer. The subject is cued to the translation targets by these objects and planes that they need to match. The orientation is cued then by the direction of that plane, and there's an audio command that tells them to open or close the hand. And so we try to sample all of these dimensions simultaneously and look at all of the different combinations of the different velocity dimensions. So we built our initial decoder mapping firing rates into velocity commands based on this automatic movement, and then we do a second round of closed loop calibration. So the term that Andy Schwartz has developed is what he calls orthoimpedance. So essentially you give the user control over the end effector, but then you attenuate any signal that is orthogonal to the direction of the intended target. And so now the person has feedback, they're in the loop, but they're prevented from making errors, and so we still get a very robust decoder. And that's what we typically use for the rest of the testing session. We do have to train our decoders every day, particularly for high-dimensional control. The first task that we really tried to characterize performance on, we're calling a 7D sequence task. So I showed you the 7 dimensions of control. This is the actual test environment that we used. So using the real robotic arm, this pegboard that we called, and each of these little circles has an LED over it. And so we're telling people where they need to move in space based on the LEDs lighting up. And the targets and success regions are just shown here in the VR environment for visualization. So there were 10 translation targets that they needed to reach to and be within that success region. And then audio cues were given to cue them as to the orientation that they needed to match and hand opened or closed. And so we tried to test all of those things in combination over time to see if they could really maintain control of 7 independent degrees of freedom. And this is the performance of our participant under full brain control. So you can see there's an audio cue that tells her to grasp. This is when we had our really terrible webcam recording these videos. But essentially, she's controlling all of the different degrees of freedom to achieve these targets in 7-dimensional space. And this is a pretty abstract task, but it did allow us a standardized way, similar to a 2D cursor pursuit or target acquisition task, to see if they could modulate across all 7 dimensions. And so in our first participant, we documented success rate. So each of these black dots is the success rate for a block of 20 trials over time. We started these experiments about a month after implant and went for another 2.5 months. And what you see is that at the beginning, her success rates were, pretty decent, so 60 to 80%, and that they increased over time. What I'm showing you in red is the simulated chance level. And so the reason that the chance level is higher at the beginning is we were using a down-weighted version of that ortho-impedance, so giving her some computer assistance to try to facilitate learning. I think what we've seen in our second participant is that's not necessarily a useful step, because we're working with people who can understand what the goal of the task is and interpret those errors. If they have decent control, we just sort of let them go with it and learn on their own rather than trying to enforce these arbitrary steps of learning. But her success rate, plateaued towards the end, 90, sometimes 100% success on that task. In other metrics of performance, her completion time for each of these blocks of trials decreased over time. She was moving more straight towards the target. We also did functional testing with her. You may be familiar with the Action Research Arm Test. It's a test of unilateral arm function. It's commonly used for people who are recovering from a stroke. And I'll show you the video of her performance. Essentially this task is to pick up and move objects of various sizes and shapes from one location and place it onto a raised surface. So this is a slightly modified version. With the largest cube, she sometimes struggled to open the hand since it was near the maximum hand aperture. She's able to position the hand down near this small cube. Her strategy was to try to scoop it up with the bottom 2 fingers. That became the term that we used for controlling those, as you'll see later. That object was much more in her wheelhouse. That was her favorite object that she liked to grab. We tested a rock. And so in all of these videos, she's controlling all 7 dimensions of the arm that I showed you before in extrinsic coordinates continuously and simultaneously. This video is not part of the ARAT, but I do like to show it at the same time because you can see how this may translate to a real-life task. So pick up the object, place it on the shelf. We would expect that she could do that. What's interesting here is that she makes sure to turn the wrist to avoid smashing the robot through the shelf, which we appreciated. And I think it's clear that her commands that she's sending to the arm are really more visualizing and thinking about what that movement needs to be, as opposed to thinking specifically, I need to move in this orientation dimension and then this translation dimension. It is a smooth and continuously coordinated movement that she's able to generate. We quantified her performance on this task. We tested 9 of the 19 possible tasks on the ARAT. There's some like picking up a washer, which I don't think any one of us could have done with that prosthetic arm. So we stuck with these that we could do in a reasonable amount of time on multiple days. Each task is scored from 0 to 3. 0 means she can't complete the movement at all. 3 means that she completed the task with normal movements in under 5 seconds. And this participant never completed the task in under 5 seconds. So most of the time she was scoring a 2. Which meant that her scores ranged typically from 15 to 17 out of a possible 27 using the BCI. Her score without the BCI, she was completely paralyzed, so her score is obviously a 0. In the stroke literature, a change of a little less than 6 points is considered clinically significant. So while this is definitely not a system that she can take home at this point, it has the potential for restoring that amount of function. Her completion times, as I said, did not become less than 5 seconds, but her average time was anywhere from 10 to 21 seconds. And just to show you, I'm not going to talk too much about the motor performance of our second participant, but his performance is similar and oftentimes better. So this is a highlight reel of his less than 5 second times that were all taken on the same day. But his strategy is much different. His is grab it and throw it on the box. So this makes it even more apparent that he's not specifically thinking about individual, movement commands. He's really visualizing what needs to happen. He's a video gamer. He's all about, scores and competitiveness, whereas our first participant was much more cautious about breaking the robot and wanting things to work well. You can see we lost a finger in that task. He's much more aggressive with it, but his control is very good. In our first participant, we were also interested in seeing if we could get different hand shapes. So, some of these may make sense to you. So pinching, closing the thumb and the first 2 fingers is something that makes sense. In order to make a whole hand grasp, we also added in something that we called scoop based on our participant strategy where, it was closing the last 2 fingers. And so when you combine these 2 hand shapes, that's a whole hand grasp versus just pinching. We did add in other dimensions, so finger, ab, and adduction, and thumb opposition and extension that our participant had independent control of, in addition to translational and orientation velocity. And initially, we started training our 10-dimensional tasks like we did with 70. So I showed you a virtual training paradigm before. That's where we ended up. But where we actually started was trying to train with a real robotic arm using this pegboard background that I showed you, where again, we're using the LED lights to cue them to a position in space. They get a verbal command of how to orient the hand and then which hand shape they need to achieve. And so you would keep doing those in order. Our feeling was that if you're going to control the robot arm, you should train with the robot arm, and that'll lead to the best control. That didn't sort of pan out, as I will show you. And so we actually moved to a virtual reality task where we were using objects, as I showed you before. So in this task, because it was 10-dimensional, we used the shape of the object to cue them as to which pre-shape they needed to use for the hand, and then they were cued when to open and close the hand. We also included, first you would reach to the target, then you would grasp the target, and then an important component of that task was holding on to that object and taking it to a new location before letting it go. So it became much more similar to the functional tasks that we were going to ask people to do with the real robot arm. And the reason that we did that is because when we had her doing 10-dimensional control, she could do the 10-dimensional version of that random sequence task fairly well. She had good control over all 10 dimensions. When we would put an object into our workspace, her control completely degraded. So when we were training with the real arm, just moving the arm in space, when we tried to do the ARAT, for example, she couldn't get the arm down near the table, or she would push through the table. There was something that was going on about trying to interact with an object that was not being captured through our calibration paradigm. And we didn't have a presentation robot to be putting objects in and out of the workspace at random locations. So we went to this virtual reality paradigm where we could incorporate at least virtual objects. And we did a small experiment to see what was the impact of that virtual reality training on performance. And so here we're looking at we called it a box and blocks like task. It's really just picking up a medium-sized block, moving it across the table as many times as you can in 2 minutes. And so on the y-axis, you're seeing the transfer rate. Higher is better. You're able to pick up that object more and more times. And here is the performance for the different calibration tasks. So this first is the sequence task, where that was how we were training with the robotic arm and no objects. Performance was okay, but not great. We did a virtual version of that sequence task, so they were not getting any feedback about objects, and it was in VR. Their performance was even less, so I think that intuition that training with the real robot did offer some benefit, but not as much as when we then added objects into that workspace. Performance was much higher. And we tested performance on that sort of random 7-dimensional sequence task performance. It was equivalent with all of the different calibration methods. And we also tested with the virtual object task. And so again, anytime we were training in VR, performance was a little bit higher than when we trained with the real arm without objects. So these are 2 videos. I'll play them side by side, where we trained decoders using our old method of using the real arm with the sequence task, and then the virtual object task doing the ARAT. And so here's some of the issues that I talked about. She can open and close the fingers, but she's just not able to bring that hand down to the table. When we trained on the same day with objects, right, she's still maybe struggling a little bit, but then is able to get down to the table, is able to issue that pinch command, and pick up that small object and bring it to the table. So we were feeling pretty good that we had maybe not understood what was going on with having objects in the workspace, but we felt like this issue was solved as far as we were concerned for BCI control. And it reared its head again, as I'll show you. One thing that I want to summarize first is, we were interested in doing these different functional grasp postures, and at least for the tasks that we were measuring, it didn't really offer her any additional performance. And in fact, so if you look at the ARAT score with 7D control and 10D control, the scores are about the same, and there was no significant difference in completion times, but they were higher with 10-dimensional control. So it was more difficult for her to do this task. I think you could see in the video, like she did reach down and pinch the cube, which is something you might do as opposed to scooping it with your 2 fingers. But aside from being able to use new strategies, it didn't actually give her any additional function. So a lot of what we've been doing recently is really just scaling back on that grasp dimension. It hasn't proven to really be an important component. I think until we're starting to integrate in sensory feedback and trying to do more dexterous tasks, a one-dimensional grasp has been fine, and they find different strategies for using that. So as I said, object interaction became a problem for us again. We continued to test with our first participant. Signal quality had perhaps degraded, and so our control, we were always sort of pushing the bounds of what we were able to achieve with the neurons that we had left. And we trained with our virtual object paradigm here. But again, you're seeing the same thing. She can't get close to the object and grasp. When she moves away, she can grasp just fine. Here, for whatever reason, she is unable to issue that close command. We sought out to try to quantify this a little bit using a really, really simple experiment where we had her reach and try to hold on to an object, or we had her reach and try to close the hand where there was no object. And we still judged on the same position targets for both of these tasks. So the precision requirement was the same. And what we saw is that when she was trying to grasp the cylinder object, the firing rate distribution skewed much higher than when she was trying to just close the hand with no object in the workspace. And we looked for individual units that were significantly changing their firing rate and really didn't see that. It became most apparent when we were looking at the entire population. This is definitely something we're investigating further, but it did become clear to us that what was happening is when there was an object in the workspace, we were seeing a correlated increase in firing rate across all of our units that didn't seem to be captured by our decoding model. This is reflected through those obvious examples that I showed you in the video, but even just the percent of successful trials when there was an object in the workspace. On this particular couple of days, she was only 50% successful. She was 80% successful when there was no object there, and our completion times for successful trials was faster when there was no object. When we look at the data, looking at the firing rate over time of the whole population, and this is standardized to what the firing rate was during the calibration session. So during the reach phase of a movement, the firing rate stays pretty low. During the advance phase, which is what we call kind of that honing in positioning phase on the object, the firing rate is much higher, and it's linearly increasing. And then during the grasp command, the firing rate is high and stays high. So what we've implemented is actually just sort of an online baseline correction for that. So we constantly take the last 300 millisecond window and compare that firing rate to what it was during training and do an online scaling to account for this correlated increase in firing rate. And when you compare her performance again, we were randomly switching between blocks where we're using our standard decoding method and this online scaled method. And you can see that here she's really struggling to get the arm down towards the table. Here, her performance is much more improved, and she's able to position the hand near the target. We've done these experiments also in our second participant, and he shows the same characteristics, and we use this online scaling for all of our testing with him as well. All right, I'm going to switch gears a little bit and talk about the work that we're doing in our current participant with somatosensory feedback. And this is work that another faculty member in our department, Rob Gaunt, has really been thinking about. So I tend to think more about the motor control side of things. He thinks more about the sensory feedback side of things. But obviously, we're working on integrating those two together. So I'll tell you a little bit about what we've seen before. So I'm sure you're convinced that sensory feedback is crucial for many tasks of motor control, right? When you're not looking at the task, to maintain posture, to prevent slip, all of those things require sensory feedback. The approach that we're taking is, again, 2 arrays implanted into motor cortex. We're recording neural firing rates, turning those into control signals for the robotic arm. At the same time, there's sensors in the robotic arm that can tell us about contact forces at different locations on the hand. We can turn that into stimulation pulses and stimulate through 2 arrays that are implanted in somatosensory cortex. This is the device that our second participant has. He's a 28-year-old male. He's 10 years post-spinal cord injury. His injury is a C5 motor, C6 sensory spinal cord injury, Asia B. So, what that presents like is he has, some biceps, tiny bit of wrist extension, and he has limited sensation on these first 3 fingers in the median nerve distribution. I wouldn't say that it's normal, but it's not drastically below normal, and no sensation on the ulnar nerve representation. This is a schematic of the device. As I showed you, 2 arrays in motor cortex, 2 arrays in somatosensory cortex. In order to minimize the number of percutaneous connections, we have a single pedestal that's connected to 1 recording array and 1 stimulating array. And we implanted 2 of these assemblies in our current participant. I'm showing you the wiring pattern here. So we have 88 motor channels and 32 stimulation channels because of the channel count limitation that's on that pedestal. Microstimulation in somatosensory cortex is not new. There's been some animal work in primary somatosensory cortex showing that stimulation in that area does evoke percepts, and that it can be used to perform tasks pretty equivalently to mechanical discrimination tasks. What we really don't know is that whether there's any training effect that accounts for this, because these monkeys are highly trained on this task, and you can only start to get this information after they've been doing the task for many months. We also obviously don't know from animal testing what microstimulation feels like. And prior to some of this work, most stimulation that had been done either in or on human somatosensory cortex through like ECOG grids has typically elicited more of a parasitic sensation. The key questions that we were seeking to answer, number 1, is, do you feel it? Hopefully, that was our first goal. This is a person with spinal cord injury. We weren't really sure necessarily if they were going to feel stimulation in somatosensory cortex. We wanted to know, where does that stimulation feel like it's coming from? How localized is it? Importantly, what does it feel like? And then does modulating the amplitude of stimulation change the perceived intensity? So if we want to convey information like grasp force, we need to be able to modulate the intensity of that sensation. For anyone who's interested, here's our stimulation. We're using traditional charge-balanced biphasic pulses, typically use a 200 microsecond pulse width. We're usually operating at about 100 hertz of stimulation. And some of these are safety requirements from our FDA approval. So the maximum amplitude that we can stimulate with is 100 microamps on up to 12 channels simultaneously. So to answer the question, where does stimulation feel like it's coming from, let me walk you through this figure. So what we did when we stimulated through each electrode is showed him this picture of a hand. And we asked him to report which of these numbered areas he felt this sensation in. And so all of these areas that we have colored are areas where he reported feeling a sensation. And so you can see we have pretty good coverage across the fingers, at the base of the fingers, not necessarily coverage of the thumb. And when you look at the organization of that, so this is the map of those 2 arrays implanted in somatosensory cortex like I showed you. This is representing the central sulcus. So this is the more medial array, this is the more lateral array. Based on what we know about the somatotopy of sensory cortex, we would expect that it proceeds from index finger to middle to ring to pinky, and that is what we see in this person who's 10 years post-spinal cord injury. Again, we did use, this is actually MEG that we used to map sensory activity prior to implantation. And so we could see that there was thumb activity located most laterally, followed by index, and then little finger, and then palm was here. What you also notice is these arrays are tiny compared to the human somatosensory cortex. And so we really had to, we tried to hedge our bets of where we wanted to place these arrays to try to get good coverage across the fingers. We did kind of nudge this one towards thumb. I would say that this seemed pretty far lateral to us, and we're trying to map sensation in somebody who doesn't really have good sensation. There was also a blood vessel here, so there are considerations that we need to take into place. But this is where we placed our electrodes. I think based on the presurgical mapping and what we know about somatotopy, this is what we might have expected to find. These responses have been fairly stable. If anything, what we're seeing is that more electrodes are eliciting a sensation. So again, these are schematics of that medial array and the lateral array. The lateral array has tended to be mostly index finger. We're getting some middle finger. This is now out to 500 days post-implantation. All of these are sensations that were elicited during a 60 microamp survey. So some other electrodes do elicit sensations at higher stimulation levels, but this is our standard metric. And on the medial array, again, you're seeing middle ring and pinky finger that kind of fills in over time. What you see is that particularly on this array, the sensation started on this one side and started to fill out towards the other. So our thought is that the array is actually settling more down into the cortex, and that's why we're able to elicit these sensations more strongly. Now, we thought we were going to answer that question of what does stimulation feel like, and I know people here are also doing similar work, and that's turned out to be actually really difficult. So we asked the participant, how natural does this feel on a 5-point scale, ranging from completely unnatural to completely natural. And 93% of the time he said possibly natural. And so that's better than completely unnatural, but it's also not informative. And I think even over time he's morphed into thinking like, well, I'm used to receiving intracortical microstimulation, and so yeah, it feels like it should feel, and that's natural. So I think this metric has become even less useful than it was at the beginning. It tends to feel like it's coming both from inside, from below the surface and on top of the surface. And the good thing is that ICMS at these levels has never been painful to him. We've asked him to describe the quality of the sensation using sort of standard terminology. About half the time he describes it as pressure. A third of the time he says that it's tingle, and then there's another quarter of the time that's either electrical or warm. That warm tends to be more like the warmth you would feel from an electrical sensation, but it's distinct enough that he's categorized it differently. So we've tried to force this out of him a little bit. What does that mean to feel like electrical? So in the areas that he's had intact sensation, we've done some surface stimulation, like you would do median nerve stimulation or something. And he says, definitely doesn't feel like that. And we're like, okay, so what kind of electrical stimulation does that feel like? And his answer is, it feels like ICMS of my somatosensory cortex, because that's, he's the only one who knows what that feels like. So we're trying to figure out a little bit more what that would be. And I think for yourselves, if you try to imagine somebody to describe what is the sensation of somebody touching your hand with a pen, but you can't say touching my hand with a pen, like what would you describe that as? It's a very difficult thing to do, actually. A few other interesting observations. Sometimes we were able to elicit sensations that he knew something was happening, but he couldn't necessarily localize it or tell us anything about the quality of that sensation. I think that's been seen. The same thing with stimulation in visual cortex. The tingle that he described was more like a vibration sensation than really kind of an electrical or parasitic sensation. And at times he's given us things that say, mechanical indentation at the base of his index finger, where he has some sensation, even if reduced, feels pretty much the same as stimulation through an ICMS electrode. So we've actually tried to do a lot of strange things to his hand, vibrating them, doing electrical stimulation, touching him with various things. That's kind of the closest that he was able to describe. Let's see if I can pause this. Maybe not. Well, so this is him describing it.
I'd say really faint touch, light touch, would be another kind of poking me with something. It's not very pokey.
Thank you.
A dull object such as a Q-tip-ish thing.
So these are the answers that he's giving us, right? And you can tell that we constantly probe him for more information, because he's sort of like, okay, I'll try to think about what, how to describe this, but, he'll say, you're poking me with something that's not too pokey. So we're trying to figure out how to actually quantify that a little bit more. The other thing that we're interested in is to see whether this sensory feedback is useful for anything, even if we can't necessarily describe exactly what it feels like. We have at least quantified that scaling the stimulation amplitude increases the perceived intensity. So this is a standard way of estimating the magnitude of stimulation, where they're asked to report the perceived intensity of stimulation on some arbitrary scale. The only rules are that it's 0 if it's not felt, it's twice as high if the sensation feels twice as intense. So this is the results from a single electrode. You can see that that's pretty linear. And same thing when we average across a number of different electrodes. Stimulation amplitude does seem to scale perceived intensity pretty linearly. We have done some online experiments where we essentially mapped the torque output from the different motors of the fingers to stimulation of channels that we expected to be index finger, middle finger, ring finger, and so on. And I'll show you this video. This is Rob, who I mentioned, who's doing most of this work. Our subject's blindfolded here.
I can show you the side.
So in the single finger discrimination, you can see, that was with no training. It's a pretty simple engineering demonstration, but those sensations are very localized, and he doesn't have to be kind of cued as to when the stimulation is going to happen to interpret those sensations. We did, warn him, hey, it might get tricky. Tried pushing on, 2 fingers at a time, and so there was some confusion, I think, about that. Whether it was index finger or middle finger, for example. But what is interesting is that it still elicits distinct sensations as opposed to feeling like a sensation somewhere in the middle. We've done some work to quantify the safety of microstimulation. We're operating sort of near limits where there could be potentially electrode or tissue damage. So this might be, the most clear thing is to show you that on our most highly stimulated electrode, we're still recording very good units. So that's, a very good sign that our electrodes are working and that there are healthy neurons in the space of that electrode. We've also quantified some detection thresholds for using electrical stimulation on areas of the hand where he does have some sensation. So again, these may be higher than what is typical, but across time, starting from pre-implant to this was 6 months after implant, there's a little bit of variability here, but overall there's no consistent changes in his innate sensation. So that's been another positive sign. As I showed you initially, the goal is obviously to close that loop. And so, mechanically, that loop is closed. We've dealt with a lot of technical issues like artifact rejection. He's using this BCI with sensory feedback to do tasks. But the challenge is that for simple tasks like the ARAT, picking up and moving blocks, it's going to be very difficult to show that sensory feedback has any impact, and you really don't need it, right? If you're looking at a cube when you're trying to pick it up, you can do that fairly well with vision. So one of the things that we've been working on at Pitt, as well as here at Case with Baloo, is trying to figure out whether we can decode grasp force related information. So, finally maybe adding something else into our decoding model besides velocity. We started with a really simple paradigm of just showing him pictures of objects that require some different level of force to maintain a stable grasp posture, ranging from marshmallow to can of soup. And we kind of tweaked these produce items in the middle so that he rated them as requiring a force level of 1, 2, 3, and 4. He felt they were evenly distributed for his own internal model of force. And our paradigm was really, really simple. We showed him the picture of what object it was going to be. He received a cue and said, now you're going to imagine grasping it. We showed him the picture, he imagined grasping it, and then there was a baseline period. We also did a little bit of a control trial where we just showed him the object with baseline trials in between, so he wasn't supposed to imagine grasping it, he was just watching it. And this is showing you the classification accuracy for the 4-object task. And so this red line is the imagined grasp condition. So as soon as the object appears, classification is still at chance level here, 25%. Once he's cued to grasp it, the classification accuracy improves close to 75% and then tends to die off towards the end of the trial. When he's just looking at the object, we don't see any modulation of force-related information. And if you look at the confusion matrix for this time right here, so you're probably familiar with this, but essentially this is showing the percentage of times that an object was reported as a marshmallow, and it actually is a marshmallow in all of those different combinations. And so what you want to see is a diagonal line here, that you're always predicting the correct object. And at least what we see is that when we tend to misclassify, we're misclassifying to the neighboring object. And so this, with a very simple experimental design, suggested to us that we could decode some grasp force information, because there have been a handful of studies that have looked at this in monkeys, but it's a really difficult task to train monkeys to do, to modulate grasp force. And so not a lot of people have actually looked at this. We've tried to do some additional work where we're using our virtual reality paradigm, where we have them reach to a target, grasp it. We tell them, hey, this sphere is now a marshmallow, tomato, orange, can of soup, and now you're going to carry it and release it. And the instruction is just to hold it with the amount of force that you need to not crush it and to not drop it. So again, this is a pretty simple first pass to see how much force information there is in there. And if we again look at the classification accuracy, during the reach time, classification accuracy is at chance. During grasp, it spikes up. For this simple paradigm, we're at 50%. During the carry or transport phase, again, we get down pretty close towards chance, and at release, it's at chance level again. Oh, I thought I had, so I guess I didn't put my confusion matrix in here. But essentially, during this grasp trial, again, when we're misclassifying to the adjacent object. That was suggesting to us, again, during grasp phase, we're getting force-related information. During the transport phase, even though it's slightly above chance, that confusion matrix is much more random. I don't think we were actually getting any force information during that transportation phase. That really suggested to us that we're decoding more of a change in force information, like wanting to increase or decrease force, as opposed to absolute force, right? Because during transport, you're still maintaining that force throughout that movement phase, but we're not seeing any discrimination across objects, at least with this very simple paradigm. We do see units that are individually tuned to these different characteristics. So again, we're showing rasters for a few different trials for grabbing the marshmallow, tomato, orange, and can of soup. And when you look during the grasp phase, you see this separation in firing rate, with the object requiring more force showing a higher firing rate. There are a few units that exhibit this response. We also see some that are tuned both to velocity and potentially force. So during grasp, you see this increase in firing, indicating wanting to close the hand. Towards the end of the grasp phase, you maybe see some separation related to force in the order that you would expect. And just to close, I'll show you just a couple slides on a collaboration that we did with Carnegie Mellon, as I mentioned. So while we've shown, I think pretty decent performance as far as reaching out and grabbing objects, there's a lot of technology out there to actually make this task easier for people and make performance much more consistent. And so, on the BCI side of things, we were still recording neural data, transforming those firing rates through our decoder into a command signal. Here we were just doing translational velocity and opening and closing the hand. What the Carnegie Mellon group had developed is this vision-guided robotic system. So there's cameras here. They're controlling this robot, so they always know the endpoint position. The cameras are perceiving the location of that object at all times, and so they know the relative position of the object and the endpoint position. So here, when you walk through this system diagram, the system perceives the object. It figures out which object it is from its object library. It knows where the hand is in space, and it tries to infer something about the intent of the person who's operating this device. So you can imagine if you're closer to the object, the system is more sure that you want to actually grab that object. And it's trying to figure out, okay, you're close to this side of the cube, so I know I need to position the hand such that it's parallel to that surface so that you would have a stable grasp posture. And then there's an arbitrator down here that figures out, how should I blend in the command signal from the user and from this intelligent control system, and then converts that into the robotic arm commands. Because you could have this task for a very simple case like this just executed automatically, but when you're working in a real environment with multiple objects, you need to have that input from the user so that they feel like they're in control of the system. The way that this task looked is that the arbitrator, as I mentioned, is looking at trying to determine how certain they are that they want to grab this object. And so this cone, essentially, is defining the percentage of computer command versus brain control command. So when it's black, it's almost all computer commands, and when it's white, it's almost all BCI commands. And so as you're moving through this initial movement, reaching out in space, it's almost completely under BCI control. When you start to enter this grasp envelope, we begin to blend the BCI control and the robot control at a percentage defined by this cone. And then during carrying the object, we're primarily back under BCI control again. For grasp aperture, there were some more explicit controls. So once you were in the grasp envelope, it would hold the hand open until you were in a position to make that stable grasp. And same thing when you were already carrying the object, it implemented a higher threshold, assuming that once you were holding it, you didn't want to drop it. So you had to issue a stronger release command. And I'll just briefly say, using shared control, for both of our participants, the ARAT scores and completion times improved. What that really looked like in terms of performance was as they were approaching the object, the endpoint speed and path length was reduced. So you saw less sort of oscillation and trying to be very precisely in the right position before issuing that grasp command. And the perceived difficulty was reduced. I'll close with this video here just showing you some steps towards system flexibility. So the system does constantly detect the position of objects in the workspace. So if the object is dropped, there's a new grasp envelope that's designed, and if the BCI user can navigate the arm to get into that workspace, the system can then assist them as it had done in the past. And here, this is a multiple object task, so that the instruction is to grab the yellow block. You can see that they kind of initially make that move towards the wood block, so the system is pretty sure that they want to grab that one. But by issuing a command that brings them out of that grasp envelope, the BCI user can take back control over the system, move towards the correct object, and take a grasp. And so this is a very simple two-object scenario, but that's what would need to be improved so that you could use this in a real-world environment with multiple objects. So I hope that I've shown you that intracortical BCIs can enable people to perform reaching and grasping movements with a robotic arm. I have let you in on some of the challenges that we've seen with object manipulation, and that this causes changes in firing rates that we had not been capturing with our decoding models. Hopefully shown you also that intracortical microstimulation can deliver graded percepts that are localized to individual fingers. And we are interested in pursuing this idea of taking advantage of existing robotics and vision technology to improve performance and stability over time. I should acknowledge the very large team of people that I work with. Mike Boninger is the clinical lead on this study. Andy Schwartz has done a lot of this work in monkeys for a number of years and really helped us get this program started. Rob Gaunt is working primarily on the sensory side of things. Elizabeth Teller-Cabarra is our neurosurgeon who does these implants. And most of this work, I would say, that's in this talk is related to our 3 grad students, Sean Downey, Charlene Flesher, and Jeff Weiss, who've really kind of pushed this work forward in our first 2 participants. So thank you. Any questions? Bob?
So it's really great work, by the way.
Thanks.
On this last. Oh, okay. For the last section, that's really interesting. But I just wonder if you really need a BCI to do that. If most of the positioning is done by the robot, like you could use a head pointer or something.
Yeah. Well, so I think for the examples that we showed here, probably not, right? I mean, I think the idea is that when you're working more in a cluttered environment and you want to know something about intention, like do you want to grab something up to move it across the table, or do I want to grab it in such a way as to pick it up and move it? That's where potentially having a BCI could use it. The reason we approach them was initially when we were having these challenges with object interaction and seeing these oscillations, and then it became difficult to stabilize the hand near the object. And so we said, if we can figure that out from vision, what is the stable grasp posture, that could actually help improve the stability of the system. There has been some work showing that people don't necessarily want an completely autonomous system, that they want to be part of the loop. Whether or not you need an intracortical BCI for that versus eye tracking or some other input depends really on the person. But, okay. Yeah.
Your second subject is a C6 Asia B, right? Is that what you said? So how much sensation does he have in his elbow and shoulder?
He has pretty intact sensation in his elbow and shoulder.
Have you noticed a difference? I don't know how to describe this, but sort of the quality of velocity recordings when you have the proprioceptive feedback from those joints?
So for him, he always leaves his arm still. He does actually have some shoulder pain, and so when he moves his shoulder, we see major increase in firing rate that it degrades performance. I think for him particularly, I don't know that it's necessarily proprioception, but it's, when he moves his shoulder, we see a really strong response. In our first participant, because she was motor complete and had intact sensation, we actually did some experiments where we had her do a 2D center out reaching task, and then we just physically picked up her arm and tried to move it into those targets, thinking that having proprioception would help. We tried blindfolding her and having her do it. It actually turned out to really hurt her performance, particularly when she was looking at it, because we were seeing, again, changes in the neural firing rates of motor cortex when she had that sensory feedback. So we went back and tried to train our decoder while she had that proprioception, and then performance was improved in that case. We tried doing some different, sort of tricky things where we would have her just move the robotic arm back and forth, right and left. When we moved congruently with that and trained with proprioception, her performance was good. She could do it without vision. With vision, it was improved slightly. But when we would move her own arm incongruently with that, she could not overcome that, even for a really simple task of just move the robot arm right, even though your arm's left. It was really dominating the activity in motor cortex there, that it needed to be congruent. So, Ron?
Thanks, Jen. I was wondering if you had any, 15 to 20 seconds is kind of a long time to complete a task, but I'm wondering if you had any indication of the cognitive investment people were making. I mean, have you looked at kind of dual tasking, or, how hard are they concentrating?
Yeah.
To do this?
So if we ask our participants, they say it's not hard at all. We've tried to do some characterization of distractions, so either having them carry on a conversation, doing mental arithmetic, doing counting tones, keep track of how many highs and lows. And when they're doing simple tasks, we can't really distract them in a way to decrease performance. Where I think it comes in, our participants, we have a lot of tourists come through the lab, they're able to shake people's hands, they don't get nervous, they can chat. It's not that cognitively demanding. What happens though is if they miss the target and need to make that small correction, that's when they need to attend and put their full attention.
So, has there been any evidence of, I know people don't do this every day, but is there any evidence of embodiment? Do people feel the arm is theirs? Is there a sense of agency with this?
I've seen it less with our current participant who actually has the sensory feedback. Part of that is the arm is positioned a few feet away from them. Our first participant, though, she named the arm Hector, and she named everything, her car, all this stuff, right? So she named the arm Hector, and when she was doing well, she was like, look how awesome I'm doing. And when it wasn't working, she's like, Hector, what is going on, right? So, and she could verbalize that, kind of looking back on her performance, is when it was working well, she felt like it was a part of her. When it wasn't, it was Hector and he was misbehaving. So I don't think it's fully embodied, but we've seen little trends like that. So, yep.
So I was curious if you looked at the issues that you showed with your first subject with adding the grasp object in. Did you look at that same training issues with the second subject now that you kind of added sensory feedback? So when you add an object, it turns it from more of a feed forward to a feedback control. Did you look at if that improved it at all?
Yeah, so we've done it a couple different ways. We initially started training our subject with the virtual object task because we felt like that's what we should do. We've gone back and tried training him without objects, and we definitely see that degradation in performance. We see that same correlated increase in firing rate. Part of our hypothesis was that maybe that firing rate is related to the expectation of sensory feedback that they're not getting. So we've then done that task with providing ICMS, and we still see that effect. So it doesn't take that away by having that sensory feedback. I mean, it's not natural. It's not fully restoring sensation. Same thing, we thought maybe it was force-related information that was being encoded, but we don't see that information until after the grasp command is initiated. So we're still a little bit mystified as to what exactly is causing that.
So this is kind of related to Ron's question, the last question. I was kind of intrigued by the degradation when they were manipulating objects. And I'm just curious if you think that has to do with a distraction, right? Having to attend to a real task versus kind of the complexity of the multi-joint control. I mean, it's sort of two different aspects.
Yeah, we tried to control for that in that experiment by still setting the position requirements for the task to be the same. So, when they had to reach to a point in space and close the hand, it was still judged as if they had to grab that object. We did have also like a point, I didn't show it here, but we did have there was a point in space that they had to be touching and do that grasp command, and we didn't see that object interaction effect. So I think that might be it, but we have not come up with the right experiment to answer that question. It's obviously some sort of abstract concept of trying to plan either a stable grasp or to grab something that you're going to use, right, as opposed to just a simple arm control, but we haven't been able to figure that out yet.
So you mentioned that you didn't have a way to put the physical objects in space, but did you try doing the physical movement but having the object within their field of view? I guess, I don't know quite if it's the perception of the object or the thinking that they're going to be interacting with the object that's doing it, but did you try having them seeing that object, even if it wasn't necessarily you were able to move it around to the different spots during the training?
I don't think we've tried that. I mean, yeah, I think in order to get a decent decoder, we really need to sample the whole position space, and so it would be challenging to have an object. We did try to have a grad student presentation robot where he was just putting objects in the workspace, and I think that was very distracting, right? So you're getting that mirror neuron response of this person moving all around, and that didn't work out so well. So yeah, I don't necessarily know if it's just the perception of that object. I mean, having it in the workspace does not inherently increase the firing rate. We've shown that. Unless you're intending to act on it, we don't see that increase in the firing rate. Whether having an object sort of in your periphery while you're training, I don't know if that would change things.
Ask a question. Do I need a mic?
It's the rules.
It is recording. So I want to go back to the cognitive load with this control. So one of the applications we're thinking about is a neuroprosthetic application in stroke population. And in particular, its population is more severely impaired. In that population, spasticity, hypertonia, especially flexor hypertonia, is really significant. And what we've experienced is that in our neuroprosthetic systems, when we try to give them control, and they are working really hard at it, the flexor hypertonia that kicks in completely overrides the electrical stimulation. And so one of the things we thought might be a solution is a cortical control. And so have you actually quantify the so-called the cognitive load, and have you ever looked at for these patients, even when they're looking at controlling a robot, looking at their hypertonia, for example, even in spinal cord population?
Yeah, for neither of our participants have had much spasticity or hypertonia, so we haven't been able to ask that question. We've talked about trying to do some EEG recording with our participants as well to get a measure of attention or cognitive load to see if there's more global changes. But otherwise, aside from asking them to rate difficulties of tasks, when we've tried to do these distraction things, it doesn't change their perceived difficulty rating. The only one that did was having them do a simultaneous motor task. So trying to attend to two different motor tasks was more distracting, or subjectively had more cognitive load.
Yeah, about the stability of the array. So the electrode, you didn't have attenuation over time with the units, the detection of the units and all of that.
We did. I mean, so I think we saw the typical, decay over the first 6 months or so of the array. But then for both participants, it had been fairly stable. Our first participant was implanted for 2.5 years. Kind of from that, 6 to 12 month period, it was fairly stable after that. Our current participant's been in for 1.5 years, and all of them, until their end point, are able to control a robotic arm.
Did you lose some units over time? Did you lose units? So
We did, yeah. Again, kind of up to that 6 or 12 month period, we were losing units.
Okay.
But then it remained pretty stable after that.
Oh.
So I don't think we could go back to do like that 10-dimensional control. That's where we were really pushing the limits, kind of at peak unit count. But as far as moving the arm in space, opening and closing the hand, we've definitely had enough units for that pretty stably. Okay, thank you all for your attention.